Good day, my name is Arul and today we are going to discuss about Barr body identification, the experiment on how to extract it and how to identify, separate it and the significance in forensic science. So behind me I have made a contraption of charts and I made diagrammatic representations, and in between we will be going through several animated videos as well and case studies in the last, and let's move into it. Initially, I want to show you the scale of this as a full experiment is represented here, and I will go through one by one. Here, we are starting with what is a Barr body. It is an inactivated condensed X chromosome found in females, and this can only be found in female individuals in case of humans as well as other beings like monkeys, chimpanzees, rats, or any type of monkeys or pigs as well. It can be only found in female, and they are the inactive X chromosomes. Whereas when we are having in males, there is X and Y, but in females there is two x's. To compensate or balance it, one of the x is getting inactivated. So let's go to the source where we are getting the Barr bodies. So Barr bodies are found in nucleus of somatic cells in female, seen during interphase of the cell division, and it is situated at the periphery of the nucleus. And if in this figure we can see that sometimes it can be here, or in the last figure it can be seen at the end of a cell. And moving on, and exactly the sources, it is in buccal cells generally attached to the envelope and neutrophils, and rod like structures called drumsticks, and in neutrophils we can see them as rod like structures. And the sources, main sources, are extracted from a dental pulp, hair follicle, blood, cartilage, buccal smears, vaginal smears, and skin biopsy. Now, biopsy is a method where we directly inject a needle-like structured machine onto the flesh and directly extract and grab the skin part out of the body, and then we analyze using microscope or any other instruments. That is a direct injecting and taking of the sample, like in cancerous cell or diagnosis of cancer. Now, use in forensic science. This is our main part. Now, relating origin of certain congenital diseases. We can find out several diseases, and we can find if the person is having a disease and if it is transferred or gained through the ancestral line. And we can diagnose, we can find out the sex of the intersexual states in prenatal as well as fetal state or postnatal, in the fetal life as a child, and after natal state we can find the sex of the individual. And in the number of Barr bodies, the whatever amount of Barr bodies that we get will give an insight about the abnormal karyotypes of an individual. And in this case we are having Turner's as well as Klinefelter syndromes as example for presence of Barr bodies in the organisms. Now, two X's are female are having one Barr body per cell. 2x female are having one Barr body per cell, and 3x having two Barr body per cell. And double XY in male is Klinefelter is present, that is having one Barr body per cell. Now the frequency of Barr bodies in case of nervous tissue is 85%, in case of amniotic and chorionic epithelium is 96% and can increase as well. And oral smears like that we are going to take in our current experiment that I am going to show will vary between 20 to 50% in normal female. Now the history. In history, Murray Barr discovered in 1949 the structure of the Barr body, and it was initially named as sex chromatin. And Edward Bertram is a graduate student of Murray Barr. And in this diagram, we can see the mouth, the tongue, and the cheek part where we are taking the buccal smears from. And these three diagrams, this I made, this is a Murray Barr. And this is a brief representation of the drumstick shaped bar body encased in the cell. And these three are the diagrams that is for the history and the bar body and Mr. Bar. Now we'll move on to the structure of bar body. Now in this bar body structure, which is a part of neutrophil, inside the neutrophil we can see the bar body. We are having the black structured heterochromatin and the white small globules heuchromatin, and we have the outer the plasma membrane, and in here this is a bar body, a drumstick appendage, and we are having the cytoplasm, that is outer wall like structure, which is the outer side of the cell, and nucleus, which is a cytoplasm. And we are having the granules from the cytoplasm, which is cytoplasmic granule, and we are having the nuclear envelope, that is actually the border of the cell here, not the outer part, that the inner part. Now this wall as a wall, these are the fragmented nucleus. And yes, and this is actually a structure from transmission electron microscope, which is in 2 micrometer, and we can see the transmitted, that is the 3D type of an internal image of the bar body structure or the cellular structure using TEM, transmission electron microscope, TEM. And this is a more detailed structure that I was able to find from the web. Now to do the procedure, it's a very easy way to conduct the procedure, and by for staining in between we can use the methylene blue as well, and we are currently using acetocarmine, but if methylene blue is available we can use it and it will give a blue color stain, and currently for acetocarmine we will be getting a red colored stain. So let's move on to the procedure.
Before going to my chart procedure that I have drawn, we'll go to what first AI has given me, and has given me some good things as well as bad things, but mostly it has given me bad things, and it is not enough to represent the bar body experiment, but it did show me in animated form as well as a realistic manner. There's two type of AI generated videos that it gave me, but it's not having any voiceovers, and that's why I'm giving the voiceover. And it has shown the exact staining, it is shown the microscopic analysis, and in humanized form it is showing the fixing of the alcohol and ether for the saliva and the acetocarmine staining and the microscopic experiment, but it is not showing the result of what we got. So this is what the AI generated for me, and I could have gotten more, but there's no confirmatory to it. So now we will be moving on to the rest of the video which I made in the chart representation, and after this video there will be more AI videos generated.
For the procedure, let's start from the mouth. From the mouth, in the female mouth or the male mouth, in that's the case, but let's consider the female mouth. In the walls of the cheek we'll be getting buccal cells. From using a cotton swab we will be collecting those buccal cells, and this smear is smeared or pasted or transferred to a glass slide which is cleaned using alcohol and cotton. After drying only we are swabbing this, we are placing the swab on the glass slide, and next we will be putting it on a equal ratio of alcohol and ether, like 50:50 ratio, like say 1 ml of alcohol, then we'll be taking 1 ml of ether, and or if it's 5 ml of alcohol we'll be taking 5 ml of ether, and this is used for fixing our buccal or a salivary buccal smear sample onto the glass slide, and it will not be removed easily after fixing it. And for 5 to 6 minutes we can place, or we can just put it dipped into the Coplin's jar. This is called Coplin jar, and after fixing it we can move on to drying, and after drying we will be using the acetocarmine in a new Coplin jar or a clean Coplin jar, and the same one, the dried one, we will be dipping it here for about 5 to 10 minutes until whatever stain is present, it will be transferred to the smeared glass slide. And then also we are drying it, and after drying it we will move on to using a dropper and a distilled water or direct running water, and we will wash slowly the stain off, where we can get a small parts of the bucket of the bar body life or the cells that we need remained on the glass slide. And then we will move on to, then after washing it is a bit dried, or we can use a cover slip and we can directly observe on the compound microscope. And after observing on the compound microscope, on in case of female cells, we will be able to observe this drumstick shaped one at the edge of the cell. And these are cell nucleuses, that's the dots, and in some cases there are two, some cases there are one only. And I mean, above one bar body, which is not very common, and we will be able to see a dumbbell shaped nucleuses, and which are called bar bodies. In case of males, we will be not able to see any dumbbell shaped bar bodies or any bar bodies at all, and we will be able to see such kind of structures. We will only be having nucleuses and cells, and next rest of part will be epithelial cells surrounding other parts. Now, why this is used? Why this is used? We already discussed this. This is used to find the sex of the individual and for identifying the human remains. There are cases where we will see that a lot of bodies are getting from any landslides or any other cases, but we will not be able to find the sex of the individual. And this can be used to find the sex of the individual, because we are not only taking the swab from the mouth, we can also take the swab from multiple locations like dental pulp, which can be remained after everything: dental pulp, hair follicle, blood. But dental pulp and hair follicle, skin biopsy cartilages, they can be remained after the body will reach a particular range of decomposition. Now, finally, we will be discussing a case study. This is a very well-known case study. I've drawn a representation by the art of the bus where the incident took place. It is a very tragic case that almost everyone in India and the world knows. This is an Nirbhaya gang rape case which happened in Delhi 2012. And this brutal 2012 Delhi gang rape case, forensic experts from All India Institute of Medical Science, AIMS Delhi, and Central Forensic Science Lab used Barr body testing using the investigation. And if my analysis and study is right, in Nirbhaya case, a lot of forensic importance or a lot of forensic techniques were used to find different aspects of the crime. So here, they used bar body testing during the investigation. The purpose was to confirm the sex of the accused individuals from biological stains. Semen sample epithelial cells which were remained on the crime scene, they were collected from the victim's clothes and body swabs. And bar body swabs analysis as sex chromatin, and sex chromatin, which I already said was the initial name of the bar body, which was also named by Edward Potron, and was found to differentiate male cells from female cells, as currently we are also doing differential extraction of semen to separate the male cells from female cells as well, the semen cells. And here the Barr body cells analysis was performed to differentiate male cells, that is male cells having no Barr body, from female cells which is having Barr body, helping to verify that the biological evidence indeed came from males. Now, this finding supported DNA profiling later on, strengthening the prosecution case. That we were able to analyze, I mean, not me, the investigators at the Nirbhaya gang rape case were able to analyze whose sample or whose excretions was what, and if the perpetrators and victims were how much involvement or how they were involved, or if they were involved. These all questions were answered using the Barr Body Test as well. They were also using multiple other tests as well to make that 100% confirm. I thank for the attention that I received during this 10 to 12, 15 minutes of video, and I'm very glad that you have listened till now. And I want to make you sure that the bar body is only seen in females, and how significant is it is in the field of forensic science in determining the sex of the individuals during the very serious crimes as well as the minute crimes for identifying the individual. And this is still used, not predominantly, but it is still used as an easy way and quicker way to find find the identity of the individual or sex of the individual from a crime. Thank you.
Gently scrape the inside of the cheek to collect a buccal sample with a sterile swab. Dip the swab into a Copland jar containing the alcohol ether fixative. Remove the swab and allow the sample to air dry. Place the dried smear slide into acetocarmine stain to color the cells. After staining, dry the slide and rinse carefully with distilled water using a dropper. Here is the prepared reddish-pink smear on the glass slide. Place the slide onto the microscope stage for examination. Under magnification, observe the cell nuclei. One shows a drumstick-shaped Barr body, indicating X inactivation.